Hey everybody, in this video we are going to talk about transfer RNAs. So in this lecture 15 this semester, we're focusing on translation, and in this video we will focus on transfer RNAs. So as we've seen in the past, transfer RNA encoding genes are transcribed by RNA polymerase III. Now, these transfer RNAs, after processing that initial transcript, the transfer RNA is going to be about 80 nucleotides long, and each transfer RNA is going to bind one amino acid. So one amino acid, and the amino acid bound by each RNA, each transfer RNA, is called the cognate amino acid, or the connected amino acid. For example, if there is a transfer RNA that has the cognate amino acid as phenylalanine, we would write it like this: tRNA with a superscript phe for phenylalanine. So this one connects to phenylalanine. So another tRNA, let's say this one connects to glycine. So we could write it like this: tRNA superscript glycine. This one, its cognate amino acid is glycine, so connects to glycine. Now, the structure of the tRNA is a cloverleaf structure. So cloverleaf. And it looks something like this. And the structure is kind of complicated, and I hesitate to put diagrams that, found on the internet, because these are YouTube videos. And I don't always know who to credit for those diagrams. And if they're copyrighted, I don't want to get in trouble for that. So I created a diagram here of a transfer RNA. So this is the typical structure. There's a 5' end and a 3' end, right? These are RNA molecules, and RNA and DNA molecules. The strands have 5' ends and 3' ends. The 5' end has a single phosphate group here. So connected, and it usually starts with a G or an A at the 5' end. And these little bonds right here, these are intramolecular hydrogen bonds. And what we're getting is base pairing, between the bases, and we're essentially getting intramolecular base pairing. So we can see a G bound to a C, G to C, and G to U. We can also get G to U base pairs in this structure. So those are non-standard base pairs, but we can get them in intramolecular base pairing. And so the intramolecular hydrogen bonds, the intramolecular base pairing, same thing. So they're going to hold this structure together. And so let's call this the stem right here. So we have a stem in like 3 loops. This loop right here is going to be called the dihydrouridine loop. Now this is a non-standard base, so after transcription of the transfer RNAs, enzymes come in and they modify some of the bases in the ribonucleotides. So there's an enzyme that modifies uracil to dihydrouracil, and then we get the dihydrouridine base. We also have a pseudouridine over here with this symbol right here. I hope you can see this in the video. Some of these symbols I diagrammed them, well, they're kind of small, aren't they? They're kind of small. So we can also get thymine in here. So thymine, so some strange bases inside the transfer RNA molecule. So this loop right here, this is called the D loop. This loop over here is called the T loop, or you can also call it the T pseudouridine C loop, or T pseudouridine C G loop. This loop down here, this is going to be called the anticodon loop. This is the anticodon loop. And this is where the anticodon is. And you can probably guess the anticodon is that thing that is matched up with the codon during amino acid synthesis. And here are the three bases that make up the anticodon in the tRNA molecule. Now, directionality with respect to the is important anytime you're talking about DNA or RNA molecules. So because of the way I diagram this, you can see 5' end over here. This is the 5' end of the anticodon, and this is the 3' end of the anticodon, because we can see the 3' end up here. So I'm going to end up flipping this around in some of the next diagrams we look at, but always pay attention to the general diagram or the context of the diagram so you can see where the 5' and 3' ends are of the anticodon or of the codon when we start looking at the messenger RNA. So I think that's it. Also, so when this is an uncharged tRNA, when the amino acid is linked to the tRNA, we say that tRNA is charged. A charged tRNA is also called an amino-acyl tRNA. And we'll talk more about that next, I believe. So briefly, before we get onto tRNA charging, let me just introduce this concept of iso acceptor tRNAs. And my pencil's getting a little dull, so it might be time to switch it out. And remember, it's gonna break probably. The tip's gonna break a little bit, but we'll be okay. We'll keep moving forward. So iso acceptor tRNAs, what are they? So they are tRNAs that recognize or have the same cognate amino acid. Iso acceptor tRNAs have the same cognate amino acid. So here are two examples. tRNA glutamine. Here's another one. Oh, well, this one, it really broke this time. Of course, I said, you know, we just keep going when it broke, but wow, it really broke. New pencil. tRNA glutamine. Okay, two tRNA glutamines. This one right here, let's say this has a codon that is GUU. This one down here, let's say this codon is GUC. So these are different tRNAs encoded by different genes in the genome of whatever organism we're talking about here. Different tRNAs, but they have the same cognate amino acid. They both bind to glutamine. So we would call these two tRNAs isoacceptor tRNAs because they both bind to glutamine. Okay, so before I alluded to charging of tRNAs, so we have uncharged tRNAs before they have an amino acid linked to the 3' end. So let me quickly diagram an uncharged tRNA. So 5' end, 3' end, no amino acid here, anticodon loop down here. Now the enzymes that charge tRNAs are called amino-acyl tRNA synthetases. And you know, I still don't know why can't we just call them amino-acyl tRNA synthases? Why do we have to call them synthetases? I don't know who came up with that, but I'm not happy with the person who came up with that. So amino-acyl tRNA synthetases. So an amino-acyl tRNA synthetase will take this uncharged tRNA and it will add the cognate amino acid to this tRNA. So if it's a tRNA whose cognate amino acid is leucine, it will add a leucine to the 3' end. We won't worry about the specific bond that is formed. We have enough bonds to keep track of. So this is a charged tRNA. In a charged tRNA, another name for that is an amino acyl tRNA. So a charged tRNA is an amino acyl tRNA, and that's where this name comes from, amino acyl tRNA synthetases. It's synthesizing an amino acyl tRNA. We can call these enzymes AATSs. Now, cells in most species have 20 AATSs, so 1 for each of the amino acids specified by the genetic code. So what does that mean? So let's go back to our example isoacceptor tRNAs. There's one, and here's the other isoacceptor tRNA for glutamine. There might be others, but we only need to worry about 2 to illustrate this concept here, that both of these tRNAs that bind glutamine, they're both charged by the same AATS, the AATS that binds glutamine to tRNAs. Which is good. That means the cell doesn't need a separate AATS for each tRNA. It could conserve resources by using the same AATS for the whole group of isoacceptor tRNAs. Okay, so I think, where did we want to, I feel like I missed something. So far we talked about, okay, charge. Now let us talk about, okay, so how are the anticodon of the tRNA and the codon of the messenger RNA matched up during protein synthesis? So let me diagram. Now this is where I start diagramming the tRNA in the reverse direction. Okay. So now I am focusing in on the anticodon, right? I'm just going to diagram those bases specifically. We kind of have a messy looking tRNA here. The loops aren't to scale, but I'm doing a pretty good job with the stems. Sometimes I draw the stems too far apart, the strands of the stems too far apart. Oh, this should be a little smaller. So the 5' end is a little shorter than the 3' end here. 5' phosphate, 3' end has the amino acid. So if this is our messenger RNA, do you remember what the cap is on the 5' end of a messenger RNA? 7-methylguanine. So somewhere down here, we have, I'm not diagramming the ribosomes yet. We're going to talk about how the ribosomes load on the messenger RNA and translate it to make the polypeptide chain to synthesize the protein. We're going to talk about that later. But here I just want to show how the anticodon of the tRNA, this is the anticodon, 3 bases, is paired up with the codon of a messenger RNA. So U pairs with A pairs with U, C pairs with G. Okay, so we have this pairing going on here, and the hydrogen bonds are forming here. Now, again, this is the anticodon. this is the codon. So the codon is found in the messenger RNA. The anticodon is found in the tRNA. Now it's very important to pay attention to the directionality here. So it has to be antiparallel, just like in DNA where the two strands are antiparallel to one another. The two strands in this pairing interaction between the tRNA and the messenger RNA must be antiparallel. So this is the 5' end of the messenger RNA and the 3' end, well, the 5' end of the codon, the 3' end of the codon. This must be the 5' end of the tRNA, and this must be the 3' end, well, the 5' end of the anticodon and the 3' end of the anticodon. So good, they're antiparallel. And how do we know this is the 5' end and the 3' end? Well, for one, we can look up here, 5' end here, 3' end here. But if we just had these bases, we would know, okay, if I wasn't showing the rest of the codon, you would know that the anticodon has to be matched. When it's paired up with the codon, has to be antiparallel. So if I diagrammed up here, the tRNA, the anticodon of this tRNA is 5' CAU, 3'. So 5' CAU. And it recognizes a messenger RNA with a codon 5' AUG. So I should probably specify tRNA anticodon matches an mRNA codon with this sequence. Anticodon with this sequence pairs up with a codon of this sequence. So why is this backwards? Again, it's the 5' and 3' ends are here, so it's just diagrammed in the reverse direction down here. Okay, so that's how different tRNAs pair up with different codons. That's how tRNAs use the anticodon sequence, the sequence of the anticodon, to recognize specific codons. So what I want to talk about next is a little bit about the genetic code, because we have a problem. And the problem is that the genetic code has 61 codons that recognize amino acids and 3 codons that recognize stop codons. Or I guess so. So amino acids and 3 codons that are stop signals. So the 3 stop codons are not recognized by transfer RNAs. Well, when we get to actually how the ribosomes perform translation, we'll talk about how the stop codons are recognized. They're not recognized by tRNAs. But the 61 codons for amino acids, well, those are recognized by tRNAs. So in theory, cells should have 61 tRNAs for each AA amino acid specifying. Is that how you spell specifying? Specifying? I have no clue. Each amino acid specifying codon. Each amino acid specifying codon. Dash after amino acid. Amino acid specifying codon. In reality, cells only have 40 to 50 different tRNAs for all 61 amino acid specifying codons. So how can 40 to 50 tRNAs, and why is it 40 to 50? Each species has a different number of tRNAs. Some species might have 40 different tRNAs to recognize all 61. Some might have 50 to recognize all 61. So how are they doing it? And the answer is this thing called Wobble base pairing. Now, Wobble base pairing was initially hypothesized by a scientist named Theodore Wobble. And I'm just kidding. Wobble base pairing was originally hypothesized by Francis Crick. And you probably recognize this name Crick from the Watson and Crick model of DNA. So Francis Crick first hypothesized the existence of Wobble base pairing, and essentially, he was pretty good at, especially for the 1950s and 60s, pretty good at, I guess, just imagining or theorizing of how different molecules of DNA and RNA fit together. And he thought, well, in a certain position in the anti-codon-codon pairing, there's probably more room here, and if there's more room, then you might be able to get these non-standard base pairing interactions there. And he came up with this idea of Wobble base pairing, and it turns out, we think this is true. It seems to be true. Okay, so what's going on in Wobble base pairing? So because of Wobble base pairing, 40 to 50 tRNAs can recognize all 61 amino acid specifying codons. So, okay, what is Wobble base pairing? Wobble base pairing. And I remember being really confused about Wobble base pairing when I was an undergraduate student 25 years ago. I remember being really confused about this. I don't know why. So let me diagram. Let's pick a leucine, a tRNA with leucine as the cognate amino acid. Okay. Okay, there's our anticodon. This is the 5' end. How do I know that? Because the amino acid is always attached to the 3' end. And let me diagram the messenger RNA. I won't put the 7-methylguanine cap. Here's the codon, the bases of the codon in the messenger RNA. Why not? I'll put the poly A tail in here. And okay, I can't resist. I'll put the 7-methylguanine cap. Now, let's say this is GAG, and this is CUC. Okay, that makes sense. G pairs with C, A pairs with U, G pairs with C. This is just the standard Watson-Crick base pairing that we've covered when we covered the structure of DNA. G pairs with C, A pairs with T in DNA, but U in RNA, G pairs with C. So the codons here, CUC, if we look this up in a genetic code, a table of the genetic code, you would see that CUC specifies leucine. So codon specifies, is that YS specifies or IES? I have no clue. I should check my spelling before I practice. Like I said, these videos are one takes. So one take, Tom. That's me. I only have time for one take, and I hope it works. So let's see. CUC specifies leucine. So if you look that up in a genetic code, you'll see CUC specifies leucine. Okay. So I haven't quite showed you what's going on with Wobble base pairing yet. This right here is the Wobble slot with respect to a paired anticodon-codon pair. So this is the Wobble slot, this position. This is, in the Wobble slot, is the 5' base of the anticodon, right? This is the 5' end. 5' base of the anticodon is in the Wobble slot. 5' base of the anticodon occupies the Wobble slot. And the 3' base of the codon. So 5' base of anticodon, 3' base of codon occupies the Wobble slot. And if we were to look at a crystal structure, I think, of this pairing interaction, we should be able to see that there's more wobble or room here for these bases to sort of wobble, They're supposed to be able to wobble relative to one another. And because of that, there are different pairs or acceptable base pair patterns in this position. When bases occupy this position, they're able to hydrogen bond differently between one another than they would if they occupied one of these positions. As a result, there are different acceptable base pairs when the bases occupy the Wobble slot. So here are the Wobble base pairing rules. Wobble base pairing rules. Okay, so in the anticodon, the 5' anticodon position, and over here we'll put the 3' codon position. Let me see, how about 5' anticodon base, 3' codon base? These are the bases that are occupying the Wobble position. So if there's an A in the anticodon, well, that can pair up with a U in the codon. If there's a C, well, that can pair with a G. So that's just standard base pairing rules right there. G can pair with C or U. So a G in the anticodon can recognize a C or U in the codon. U in the anticodon position can recognize A or G. So what's going on here? So essentially, G can recognize C, we already knew that, but now it can also recognize U. So if we put U here, we already knew U could recognize A, so it can also base pair with G over here. And then we have one more, which I'll talk about more in a second. So I, this is inosine. Essentially, if you have an adenine ribonucleotide at the 5' anticodon position, an enzyme can come over here. It can take out the, or change the amine group in the adenine and turn it into a, I think a carbonyl group. I might be wrong on that. Don't quote me on that. It doesn't matter. We don't need to memorize structures. But anyways, the enzyme can change A to I. And as a result, this non-standard base here, it can pair with A or C or G. Let me check that. I want to make sure that's right. For some reason I'm thinking sorry, 1 second. Be back in. I knew that was wrong. A or C or U. Okay, so what that means is, you know, we can get non-standard base pair interactions here. For example, okay, so this means GAG, this tRNA with the GAG can also recognize, let's take a look, CUU. So the single tRNA with a GAG anticodon can recognize CUC and CUU. Right? Because we see the 5' base of the anticodon is a G. We can look down here, 5' anticodon base is a G. We can recognize a C or a U in the 3' for the 3' codon base. So C or U. Now, for all this to work out and for translation to make sense, CUU better recognize leucine. A CUU codon, you know, must specify leucine. Otherwise, you know, this tRNA that is attached to leucine, this tRNA recognizes CUC and CUU codons. It must recognize, I mean, these have to specify the same amino acid. Otherwise, this whole system wouldn't work, and we'd be ending up with proteins that, you know, have the wrong amino acids in them. But it does. If you look at the genetic code, the table of the genetic code, you see, yep, indeed, CUC and CUU both specify leucine. So this is how Wobble base pairing can allow this single transfer RNA to recognize two different codons. And like we said before, I think in the previous, so I still have that. So this is how cells can use 40 to 50 tRNAs to recognize all 61 amino acids. This is just an example of how one tRNA can recognize 2 different codons, CUC and CUU. Did I say that? 40 to 50 tRNAs for all 61 amino acid specifying codons. Okay. And this is an example of how a tRNA can recognize 2 different codons. So now I think I wanted to talk a little bit more about inosine. Okay, so what is that inosine? Again, the enzyme that changes A's to I's is adenosine deaminase. And so here's an example. So C, G, A. So let's say, I didn't diagram this that well. That's terrible. Let's try it again. Okay, I want to do a tRNA here. Okay, P, and I think what I want to put here is C, G, A, and this is going to be tRNA alanine. So alanine is the cognate amino acid, AGC. And if we were to look this up in, well, let me draw the messenger RNA first. So 5' cap, poly A tail. So without making any changes, CGA will recognize, well, AGC, right? The anticodon. 5' AGC, 3', 5' AGC, this is the 5' end, this is the 3' end, will recognize the codon 5' GCU. Now, GCU specifies alanine. So if you were to go to a table of genetic code, you should see GCU specifies alanine. And okay, that makes sense that this is a tRNA alanine, has the codon, anticodon AGC that it would match here. Now, the enzyme adenine deaminase, it can come in here, it can change that A to an I. So let me just, I'm diagramming this part here. I'm just doing this part right here. We're going to change that A to an I. Now, because of the Wobble base pairing rules, right? If you can go back, you can flip back the video a little bit, but what we said was that an I in the 5' anticodon position could pair with A, C, or G in the 3' codon position. So this can recognize 3 different codons. So this is the 5' end, and this is the 3' end of the anticodon. These are the codons down here. This is a codon down here, 5' end, 3' end. This would be GC. Well, I can pair with U. Whoop, hold on a second. No, I can't. Let's look again. AC or G. Oh, no. AC or U. Sorry, did I screw that up again? How do I fix that? There's too many notes here. AC or U. So yes, I can pair with U. Or this tRNA with an inosine here, it can recognize GC. Let's go back again. A. Or It can recognize G, C. Let's see, what's the other one? C. Okay, now in the wobble position, inosine here can lead to the recognition of this codon here, this codon here, and this codon here. So if we were to look in the genetic code, we should see, and we would see, I'm not even going to look, that's how confident I am, that all of these codons here specify alanine. So this is an example of how the wobble effect combined with changing adenine to inosine can allow one tRNA molecule, one gene, to encode a tRNA molecule that can recognize all three of these codons. Okay, so that's how the wobble effect allows cells. We can find that again. Where's my notes? You guys can back up. That's how the wobble effect allows cells to get by with encoding only 40 to 50 different tRNAs for all 61 of the amino acid specifying codons. So I hope you understand how the wobble effect works. Again, like when I was an undergraduate, I was very confused about the wobble effect, but I think it's pretty straightforward. And in the next video, we are going to take a look at how the ribosomes actually synthesize the protein. Now that we have all the pieces, you know what, we have to talk about ribosomal RNAs first, but then we'll have all the pieces, we'll have the tRNAs, the ribosomal RNAs, the peptide bonds, we've covered all that stuff, and we'll be able to see how ribosomes synthesize peptides during translation of messenger RNA. Okay, see you in the next video.